Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from the Department of Biochemistry, Independent Medical College, Islamabad. This is my third lecture regarding the DNA replication and repair. In my previous lecture, I discussed the mechanisms of prokaryotic DNA replication. In the eukaryotes, the replication is more complex but involves the same types of mechanisms. Here in the slide I will discuss the differences between the DNA replication of prokaryotes and eukaryotes. In the prokaryotes, as their genome is of small size, a single origin of replication is sufficient in order to replicate all of their DNA. In the eukaryotes, the molecular size of the DNA, it is great, they are large in size, and so multiple origin of replications they are needed. DNA polymerases, they are DNA polymerase 1, 2, 3 and 4 in the prokaryotes, and in the eukaryotes they are the alpha, beta, delta, epsilon and gamma. In the eukaryotes there is the mitochondria, and DNA polymerase gamma is needed for the mitochondrial DNA, which is not applicable in the prokaryotes. In the prokaryotes there is no repair mechanisms, and in the eukaryotes there is a full-fledged and well-established repair functions. Theta structure is present when the origin of replication is open up. It forms a theta structure, which is not present in the eukaryotes. In the prokaryotes, obviously few accessory proteins are needed, and in the eukaryotes, due to its complexity, many accessory proteins they are needed. Only RNA as a primer can be used. In the eukaryotes, both RNA and DNA can be used as primer. RNA is the primer is removed by the polymerase 1, but in the eukaryotes the primer is removed by RNase H and flap endonuclease 1. In the prokaryotes, as there are no histone proteins, so only unwinding will be needed in order to initiate the DNA replication. But in the eukaryotes, first the histones will be separated, and after the unwinding, then the eukaryotic DNA replication will be started. This is a picture of eukaryotic cell cycle. The events surrounding the eukaryotic DNA replication and cell division, they are coordinated, and this coordination is in the form of cell cycle. The period preceding the replication is called the G1 phase. Following the DNA synthesis, it occurs in the S phase. After the S phase, S is for the synthesis. After the S phase, there is another gap, G2 phase, then is the M phase. G1 phase, and the G1 phase, the RNA and protein, they are synthesized, but no DNA is synthesized. In S phase, the DNA, the amount of the DNA, it doubles, and RNA and proteins, they are also synthesized in S phase. Another gap 2 phase, here again no DNA synthesis is present in the G2 phase, but RNA and proteins, they are synthesized. In the M phase, mitosis, nuclear division, and cytokinesis, cell division, it yields 2 daughter cells. Cells that have stopped dividing, such as mature T lymphocytes, they are said to have gone out of the cell cycle into the G0 phase or quiescent phase. Such quiescent cells can be stimulated to re-enter into the G1 phase to resume the division. There are also several checkpoints here in the cell cycle. You are seeing that there are several checkpoints which will not allow the cell to enter into the next phase unless these checkpoints they are satisfied. So if the genome is incompletely replicated, or if the normal chromosomal segregation machinery is incomplete, or there is some other problem in any type of the event, these checkpoints will not allow the cell to enter into the next phase. If the damage is repaired, if there is the damage, the cell will try to repair it, and it will enter into the next phase. But if the damage cannot be repaired, such cell undergo a programmed cell death called apoptosis. All the eukaryotic cells, they have the gene products that govern the transition from one phase of the cell cycle to another. So what are that gene products which govern the transition from one phase of the cell cycle to another? They are the cyclins and cyclin dependent protein kinases, cyclins and CDKs. Cyclins are a family of proteins whose concentration increases and decreases throughout the cell cycle, and these cyclins, they turn on at appropriate time different CDKs, cyclin dependent protein kinases. As the name shows, these kinases, they phosphorylate the substrates essential for progression through the cell cycle. Many of the cancer causing viruses, they don't have the control, or they don't have the active cyclins or cyclin dependent kinases. They either they will have excessive production of cyclin, or they will have loss of specific CDK inhibitor, or there is some problem in production or activation of cyclin and CDKs. So if they have lost the control over the entry, loss of control over these cyclins and CDKs, they will be improper, or there will be uncontrolled production of protein and cell division. Many of the cancer causing viruses, oncoviruses, and cancer inducing genes, oncogenes, they are capable of alleviating or disrupting the apparent restriction that may control the entry of mammalian cell from G1 into the S phase. So the control between the G1 into the S phase, it is lost in many of the viruses and cancer inducing genes. This picture shows the schematic illustration of the points during the mammalian cell cycle which indicated the cyclins and cyclin dependent kinases, or they are activated and when they are activated. The first of all, cyclin D. Cyclin D, its level rises late in the G1 phase. Cyclin D level, its level rises late in the G1 phase. The D cyclins, they activate CDK4 and CDK6. CDK4 and CDK6. The cyclin and CDK complex, they are the active serine threonine protein kinases. They will when the cell, it comes late in the G1 phase, there is another protein, cyclin E. This cyclin E and CDK2 and cyclin E, they form the complex also late in the G1 phase. After entry into the S1 phase, or just before entry into the S1 phase, the cyclin E is rapidly degraded and allow the cell to enter into the S phase. Now the CDK2, it will form the complex with cyclin A. This sequence is very necessary for initiation of the DNA synthesis in S phase, and this is the complex. Now the cell will enter into the, after the S phase, the cell will enter into the G2 phase. A complex between the cyclin B and CDK1, The complex between cyclin B and CDK1 is the rate limiting for the G2 and M transition in eukaryotic cell. And here again the cyclin A and CDK1, they also form the complex. So these cyclins and CDKs, they are the checkpoints which will check that whether the nuclear DNA is completely replicated or whether is there some problem in the DNA segregation, chromosomal segregation. The nuclear DNA is completely replicated once and only once. Once the chromatin has been replicated, it is marked so as to prevent its further replication until it again passes through mitosis. This process is called a replication licensing. And all this restriction points, they run in a very controlled manner. This is the table which shows the cyclin and cyclin dependent protein kinases, which kinases they are activated by the cyclins and what are their functions. I have discussed it already. So let's move on to the next slide. Okay, this is the five different eukaryotic DNA polymerases. 5 different eukaryotic DNA polymerases. They are of high fidelity, and the first one is polymerase alpha, DNA polymerase alpha. It is a multi-subunit enzyme. One subunit, they has the primase activity. Primase activity will initiate the strand synthesis on leading and at each beginning of each fragment of lagging strand. And this primer or this primase subunit synthesize a short RNA primer, which is extended by another subunit of DNA polymerase alpha, whose activity is 5' to 3' polymerase activity. So they generate short piece of DNA. Now the next is polymerase beta. Polymerase beta is involved in gap filling in DNA repair. Then is the polymerase gamma. Polymerase gamma replicate mitochondrial DNA. Polymerase delta, the delta elongate the Okazaki fragments of the lagging strand. And polymerase epsilon, this polymerase epsilon is used to complete the DNA synthesis on the leading strand. The polymerase delta and epsilon, they both have also, in addition to 5' to 3' polymerase activity, 3' to 5' exonuclease activity is also present, which will proofread the newly synthesized DNA. Telomeres. Telomeres, they are complexes of DNA plus proteins, which is collectively called the shelterin. Non-coding DNA, this telomeres, they contain non-coding DNA, and they are present at the end of linear chromosomes, only in the eukaryotes. It has several functions. It maintains the structural integrity, prevent the attack of nucleases, allow a repair mechanism to distinguish between the true end from the break, and also it prevent the end fusion with neighboring chromosomes. What is its structure? Its structure is that what are the nucleotide sequence of the telomeres? They contain A, 3 G's and 2 T's, A, G, G, G and TT. And obviously their complementary regions will contain C's and A's. GT strand is longer than its CA complement. This single stranded DNA fold back on itself, forming a loop which is stabilized by proteins. This is a schematic picture of telomeres. A young chromosome, it has a longer telomeres, and after every cell division it becomes shorter. So the old chromosome will have the shorter telomeres. This is a micrograph of a chromosome which shows that the DNA, it contains telomeres at the end, and this is the sequence of the nucleotides which might be present at the end of chromosomes. Telomere shortening. Eukaryotic cells have faced a special problem in replicating the ends of their linear DNA. Following the removal of RNA primer from the extreme 5' end of the lagging strand, there is no way to fill the remaining gap with the DNA. Consequently, in most of the somatic cells, the telomeres, they shorten with each successive cell division. But this shortening does not happen in the germ cells, in the stem cells, as well as in the cancer cells. But in these cells, in the cancer cells and in the germ cells, there is a protein called the telomerase. So due to the presence of telomerase, the telomere shortening does not happen or does not occur. In the somatic cells, the telomerase concentration is very less. Beyond some critical length, after several cell divisions, the cell will not able to divide again. This is called the senescence. Telomerase, they are complexes containing protein, reverse transcriptase, which is called the TERT, and a short piece of RNA, TERC. Reverse transcriptase uses RNA template which is present inside its structure to synthesize the DNA in the 5' to 3' direction, extending already longer 3' end. Then, after the function of telomerase, routine primase will use this extended GT strand, GT region as a template to synthesize RNA primer, and which is further extended by the DNA polymerase. I'll show you diagrammatically how these telomeric ends, they are preserved by the telomerase, and they are not shortened up in the cancerous cells or in the germ cells. First of all, see that the 3' end of the red strand at the top left side, the 3' end is longer. This end will be further elongated by the telomerase, and then after its working of the telomerase, the primase will extend the second strand. So the first step is that this telomerase, it will extend the 3' end of the DNA, and here you are seeing that it is using the RNA template for the strand synthesis. Then RNA primer is synthesized by the primase. This telomerase is removed because it has completed its function. It has extended the already longer 3' end. Now the 5' end, it has to be extended, which like in the routine started from the RNA primer made by the primase, and then there will be the function of DNA polymerase, which will use the 3' end of the primer, and this now the 5' end is also extended. This picture I took it from the Wikipedia, and I thought this is more explanatory as compared to the picture in the Lippincott. This green color ball, it is a protein telomerase, and this is its RNA primer, which RNA strand which is used as a template for the synthesis of 3 prime, for the elongation of 3 prime end of the DNA. After its extension, it is removed and further work and another strand, the complementary strand, is elongated by the DNA polymerase. Telomeres, they are the mitotic clocks. Length is obviously inversely related to the number of times the cells have been divided. So we can predict that how much will this person will be have its age. So there is an active research regarding the telomeric ends and predict that how much years this person will live. So it provides insight into the biology of normal aging. Progerias is a disease of premature aging in which the child age fast, and the cancers. As seen with the telomerase, the same enzyme reverse transcriptase. The reverse transcriptase is the enzyme which are used by some retroviruses. Reverse transcriptase, they are involved in the replication of retroviruses, for example HIV. These viruses, they carry their genome in the form of single-stranded RNA. Following the infection of the host cell, the viral enzyme reverse transcriptase uses the viral RNA as a template for 5 prime to 3 prime synthesis of viral DNA. So viral DNA is synthesized using DNA as a template, and enzyme is reverse transcriptase, which this DNA is now become integrated into the host chromosomes. Reverse transcriptase is also seen in transposomes. The transposomes, they are the DNA elements that move about the genome. These transposomes, they are first transcribed to RNA, then the RNA is used as a template for the DNA synthesis using the same enzyme reverse transcriptase, and then this DNA is then randomly inserted into the genome of the mammals. HIV kills many of the cells it infects, principally T lymphocytes, rather than causing tumor. This gradually leads to suppression of immune system in host organism. The reverse transcriptase of HIV is even more error prone. It means that every virus which is from its parents, they are different. Its gene, they are mutated in a high rate. That's why their vaccines, they cannot be formed, because there is no mechanism, there is no proofreading mechanism for the removal of the errors. So these viruses, retroviruses, they are more error prone. But there is a strategy which has been adopted by the scientists, that they made the vaccines which consist of 2 or more coat proteins of the viruses. These only coat proteins, they are not infectious, but they will stimulate the immune system to recognize and resist the subsequent viral invasions. There are many disease caused by the RNA viruses. For example, nowadays a common pandemic, this is COVID-19, caused by the coronavirus, hepatitis C, E, polio, rabies, common cold. They are all these are the diseases caused by the RNA viruses. So students, this is my third lecture. Best wishes and good luck.